In this video we will discuss the key issues to consider when planning an in vivo fluorescence imaging experiment. First, specificity and kinetics. It's important to understand that fluorescent probes will have some drug-like properties. They need to circulate throughout the body and accumulate or activate at the site of disease. They also need to clear from the body, which can present off-target signal, usually in the kidneys, bladder, and liver. These pharmacokinetic and dynamic properties will differ depending on the probe structure and chemical composition. This means that when you image a tumor, for example, you may see some predictable non-tumor sites of signal, generally at lower intensity. If you are developing your own imaging agents by using a kit to label proteins or cells, it is very important to assess targeting and specificity in vitro before commencing in vivo studies. If you are unsure how to do this, please see our previous in vitro fluorescence training video. You will also want to identify the optimal imaging time via kinetic analysis and any off-target probe accumulation or metabolism before starting large-scale studies. The second issue to consider is tissue absorbance of light. Oxygenated hemoglobin will absorb visible light most strongly at wavelengths below 600 nanometers. This will serve as a major impediment to light transmission through tissues. In our experimental model, we will image agents that absorb and emit in the far red region of the visible spectrum, so interference from hemoglobin absorption will be minimized. However, green fluorescent reporters such as GFP and FLI will have significant overlap with and interference from hemoglobin absorption. Such reporters will be limited to very shallow imaging through tissue at best. The third issue is autofluorescence from tissue and chow. Tissue autofluorescence is produced when endogenous fluorophores within the animal are excited. Autofluorescence is highest at wavelengths below 650 nanometers and reduces with increasing wavelengths. Autofluorescence background signal will be lowest in the far red region of the spectrum. Chow autofluorescence is a result of chlorophyll absorption and emission. Alfalfa-containing diets are high in chlorophyll content. Chow autofluorescence is most intense between 650 and 700 nanometers. Specifically formulated alfalfa-free diets can be used to minimize chow fluorescence. In short, for the best success for your in vivo fluorescence imaging studies, choose probes in the far red region of the visible light spectrum and switch animals to an alfalfa-free diet prior to imaging. Our living image software features tools such as spectral unmixing that facilitate autofluorescence removal. Spectral unmixing is a useful tool, but requires more time for image capture and analysis. With appropriate planning, you can sometimes get around using spectral unmixing if the desired probes have sufficient spectral separation. In this video, we will perform two experiments using the same cohort of animals based on the results of the previous video to demonstrate the use of the instrument with and without spectral unmixing. We will begin with the most streamlined imaging scenario, utilizing two near-infrared imaging agents with sufficient separation, approximately 70 nanometers, between emission peaks. Specifically, IVIS pancathepsin 680 and IVIS integrin receptor 750. Imaging will be performed in animals on a low fluorescence diet to minimize unwanted gastrointestinal signal. Let's discuss experimental design. Imaging fluorescence activity in deep tissue sites, such as the lungs, is challenging as a starting point for assessing the utility of fluorescent agents for in vivo imaging. Therefore, to test specificity of our near-infrared agents with this tumor line, we will start with a simpler model to optimize our parameters before moving into a deep tissue model. It is crucial to set up the appropriate test and control groups of mice. The use of appropriate controls helps you to confirm the target and non-target signal in your experimental animals. Controls are the key to a successful in vivo fluorescence imaging experiment. Always include positive, negative, and naive control animals for your experiments. Let's take an oncology example. Positive would be a mouse with tumor and injected probes. Negative would be a mouse with no tumor but with injected probes. Naive would have neither tumor nor injected probe. Careful comparison of positive and negative control mice helps us understand the role of any non-target related signal. This could include tissue signal related to metabolism or clearance of the reagent. Now that we've discussed the control groups, let's move on to the actual experiment. Our goal is to image the biological activity involved in 4T1 tumor progression using fluorescent approaches. Therefore, we will perform fluorescence molecular imaging of two near-infrared agents using epi-illumination in vivo. We will use epi-illumination as the source location is shallow. For deep tissue imaging, we typically employ transillumination. We have previously determined from our in vitro characterization studies that the two agents we will use in this study specifically target 4T1 tumor cells. Our first near-infrared reporter, IVIS pancathepsin 680, is an activatable agent that detects cathepsin activity within these tumors and is a 680 nanometer imaging agent. Our second probe is IVIS integrin receptor 750. It is a targeted agent that binds to cell surface alpha V beta 3 integrins and emits at 750 nanometers. We will inject these agents intravenously through the tail vein and image 24 hours post-injection as suggested in the product datasheets. We previously mentioned the importance of control mice. We will include 3 positive control animals to demonstrate the lack of overlap between the probes by having 2 animals with a single probe injection and 1 with a mixed probe injection. Therefore, our experiment will be set up as follows. We will have 5 animals. Mouse 1. A naive control bearing a subcutaneous mammary fat pad 41 Luke 2 tumor, with no probe injection. Mouse 2. A negative control lacking a tumor but injected with both probes. Mouse 3. A positive control, bearing a tumor and injected with only the IVIS pancathepsin 680 probe. Mouse 4. A positive control, bearing a tumor and injected with only the IVIS integrin receptor 750 probe. Mouse 5. A positive control bearing a tumor and injected with both probes. With epi-illumination, you can use the high-throughput field of view for image capture. This is a fast and simple process that will yield information on both cathepsin and integrin expression in all 5 animals at once with the acquisition of 2 images. 24 hours post-injection, prepare to take your images. Open the Living Image software. Click Initialize and wait for the camera to cool to the operating temperature of -90 degrees Celsius. Click on the imaging wizard. Choose fluorescence, filter pair, and make sure that epi-illumination is selected as your mode of illumination. As a default, the wizard will set up acquisition for 1 probe. To add a second probe, click Add on the right-hand side. Click the drop-down menu under the Name section to select your probe from a list or manually input the excitation and emission maxima. The alphabetical category has nearly 100 common reporters pre-programmed into the software. Lastly, choose your imaging subject, acquisition settings, auto or manual, and field of view. Click Next. The sequence editor will open. You will see 2 rows listing the acquisition settings for each probe. Click Acquire Sequence. Label the images appropriately and click OK. Once the instrument has finished acquiring the images, the status light on the instrument will change to green. Remove the animals from the instrument and return them to their cages. Now that image acquisition is complete, we can begin analysis. Our animals were positioned in the instrument as described in the setup. To remove instrument noise, apply the Adaptive Fluorescence Background Subtraction. The tool is located in the Tool palette, under the Corrections tab. Adjust the threshold until a purple mask covers the animals. You can also right-click, select Crop Area, and click and drag a rectangular mask over your subjects. Click Set when done. For quantitation, change the unit to radian efficiency. Visually, you can see the filter pairs used to capture the 2 probes are spaced sufficiently. You can distinguish individual probe signal intensities with little overlap or bleed-through signal. However, we need to determine this quantitatively. Therefore, we will generate ROIs and compare the resulting data to background values. We will generate ROIs to closely match the outline of the tumor using the photograph as a guide. You can make the fluorescence transparent by decreasing the opacity of the overlay under Image Adjust in the Tool palette. Place an ROI into the image. Click and drag it over the tumor area, starting with the control animal on the left. After resizing and moving it appropriately, right-click the ROI and either copy and paste or duplicate ROI to create a copy for the next tumor. Continue this process in the next animal. The second animal does not have tumors, so we will drag the ROI over a representative area on the chest. Continue for all animals. To better localize focal points, you can change the minimum and maximum in the color scale. These settings are found in the Tool palette under the Image Adjust tab. You can also determine where neighboring signals begin to overlap, in this case with the liver. Often probes can also be detectable in tissue surrounding tumor, as they are markers of inflammation. So, it is best to size ROI slightly larger than the tumor. Remember, we are monitoring the disease state as opposed to targeting tumor cells specifically. If an area is difficult to isolate with predefined shapes, a freehand ROI tool is available. Select Contour ROI under the ROI Tools tab and choose Free Draw. Outline the tumor by left-clicking points around the perimeter, and then do a single right-click to close the shape. Since the mice are in the same position for both filter pair images, the same ROIs from the first image can be used for the second. Go to the ROI Tools tab where it says Save ROIs. Type in a name and click Save. Now open the second image, choose the ROI template you just saved and select Load. They will now be loaded onto the second image. The Measure ROIs button will display the measurement values for both images. In vivo fluorescence images will contain a percentage of nonspecific signal from autofluorescence. Therefore, we will want to determine the average signal per area of ROI. Dividing by the area will be important, especially if you do not draw the ROIs the exact same size. So the unit for quantitation will be average radiant efficiency. The data can then be copied into Excel, as previously demonstrated. We can look at mouse 3 and 4 to determine if there is any bleed-through of signal from the 680 probe into the 750 filters and vice versa. In this example, neither probe shows a significant amount of bleed-through. This result was expected as the two probes are separated by at least 70 nanometers. Specifically, mouse 4 had near background radiant efficiency at 8.32 times e to the 7 when using filters optimal for IVIS pan-cathepsin 680, and mouse 3 had near background radiant efficiency at 8.28 times e to the 7 when using filters optimal for IVIS integrin receptor 750. Furthermore, both probes showed a high degree of specificity for the target 4T1 tumor cells. Our results show little signal in the uninjected tumor-bearing mouse number 1. This result tells us there is very little tissue autofluorescence signal captured using the near-infrared filters. There was low-level expression in the negative control mouse number 2, which represents nonspecific probe accumulation. Tumor signal at 680 nanometers is only seen in mouse 3 and mouse 5, those that received an injection of the IVIS pan-cathepsin 680 probe. Tumor signal at 750 nanometers is only seen in mouse 4 and 5, those that received an injection of the IVIS integrin receptor 750 probe. Mouse 5 illustrates the ease of capturing both integrin expression and cathepsin activity in the same tumor-bearing mice. There is low-grade systemic signal, which is expected for a systemic agent, and some bladder clearance for these agents. The tumor sizes and location are in strong agreement with bioluminescence imaging performed earlier. Yet, these fluorescence agents provide additional biological information. Quantitative analysis provides good agreement with the tumor imaging. But, it is important to note that the signal magnitude by bioluminescence imaging would not be expected to correlate perfectly with the near-infrared fluorescence. Bioluminescence provides readout on tumor burden, whereas fluorescence provides biological marker quantification. As you can see, careful experimental design will allow for extremely fast and accurate estimation of underlying molecular activity, largely circumventing issues of background signal and crosstalk. There will be some situations, however, where background is high or where significant wavelength overlap between probes causes crosstalk. In the next video, we will discuss how the spectral unmixing tool can help in those situations. In this video, we have shown how the Living Image software can image two near-infrared fluorescent probes in the same animal, using different excitation and emission filter pairs. We have also discussed the necessary positive, negative, and naive controls that you should include for any fluorescence in vivo imaging experiment. Continue to the next video to see how spectral unmixing can be used to isolate overlapping fluorescent signals coming from two or more sources. Thank you for watching.